everyone, and welcome to today's webinar, Integration of Affinité's SPR Technology for Quality Control in AAV Bioproduction. I'm Cassie Saltman of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Affinité Instruments. To learn more, visit www.affinitéinstruments.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box on the left and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. In addition to submitting questions, we encourage you to participate in 3 poll questions after the presentations. We will prompt you to record your responses to the questions when they're available to you. I'd like to now welcome our speakers for today's webinar. Dr. Marie-Eve Paquet, Adjunct Professor, Cerveau Brain Research Center and Université Laval, who will start with an introduction to AAV and advances in using SPR for AAV characterization. Dr. Alina Venereo Sanchez, CEO, V-Vector Bio, who will discuss scalable production of AAV in suspended cell and potential of SPR in the bioprocess. And Dr. Zhao, Application Specialist, Affinité Instruments, who will deep dive into Affinité SPR technology and integration in AAV bioproduction. For a complete biography on our speakers, please visit the Speaker Biography tab on the top left of your screen. Dr. Paquet, you may now begin your portion of the presentation.
Okay, thank you for the introduction, and thanks everyone for being here. Today I'm going to talk about the Canadian Optogenetics and Vectorology Foundry and the viral vector core and how we've been using SPR for our technologies. So basically, we're a lab that designs and produces viral vectors that are modified viruses that we use to deliver genes into target cells. Basically, this is a strategy that exploits the concept by which all viruses bind to cells, enter cells, and release their genetic material. Viral vectors are modified viruses that are non-pathogenic, so all the pathogenicity genes have been removed, and they can be used for research in vivo or also for therapeutic application. So on the right is an image that shows different applications for gene therapy with a type of viral vectors called Adeno-associated viruses. And gene therapy can be used for gene replacement therapy, for example, in which defective genes can be replaced, or we can also use gene therapy to introduce new genes to give cells new functions, for example. So adeno-associated viruses, or AAVs, are small viruses with a capsid of about 25 nanometers. They're quite unique in that they require a helper virus for their replication. Even in nature, they don't replicate on their own, so they're not pathogenic at all. They don't cause disease in humans. They have a linear single-stranded DNA genome of about 4.7 kb. So the packaging capacity is about 4.7, which means it's fairly small, so we can introduce only fairly small genes in there. The wild-type genome is composed of inverted terminal repeats, which are essential for packaging, and then rep and cap sequences. And to package these AAVs in the lab, we split the natural genome into 2 plasmids. One contains the packaging sequences, the ITRs, and the gene of interest, which here is indicated by a promoter followed by a cDNA. And we also need to provide the rep and cap genes. We also need a 3rd plasmid, which is the helper plasmid, which contains sequences from the helper virus, which I talked about earlier. And we put these 3 plasmids in a cell line. And so basically, we use the cellular machinery to package these viruses, and then we purify the viruses. So AAVs, it's a type of virus, but they exist in multiple serotypes. And serotypes are sequences in the capsid or different in capsid sequences that will influence binding to cellular receptor and thus influence viral tropism. And there are natural serotypes that exist, but they can also be engineered so that the virus will acquire desired capabilities, such as crossing the blood-brain barrier, for example. So here you have a list of natural serotypes from AAV1 to AAV9 and their primary receptors. So the receptors are different, which means that the different serotypes will bind to different cells on which these receptors are expressed, and different engineered serotypes. Sometimes we know the receptors, sometimes we don't know the primary receptors. So our group at Cerveau is part of the Canadian Optogenetics and Vectorology Foundry. We're the basic viral vector core. We're located in Quebec City. It's a group of about 12 research associates or professionals, and we do a lot of research and development also. So we have students and postdoc. We do a lot of production, characterization of viral vectors, such as AAVs that I'm talking about today, but also lentiviruses and rabies viruses. We also do a lot of molecular biology, tools customization. We're interested in biomanufacturing, quality control, and we're definitely the largest academic viral vector platform in Canada, and we're distributing a large number of viral vectors every year all over the world. So this is a picture of our AAV purification process. And so, as I mentioned before, it starts with a triple plasmid transfection in hex cells. In our lab, we mostly use adherent cells. You will see with Alina that when one is interested in scaling up, suspension cells can be used, but we are more equipped for generating many small preps rather than a really large prep. So we use adherent cells. So after transfection, we collect, we concentrate, and then we purify the virus through a density gradient. And then we need to analyze the vectors that we've packaged. So throughout this process, we are interested in introducing new quality control steps that could help us decide if we should go on or abort a certain run. These methods need to be fast. They need to be compatible with the presence of contaminants, for example, for in-process analysis. And another thing that we'd like to do is determine the ratio of empty versus full capsids. So at the end of the process, you'll see here on my figure that most viruses actually contain a genome, but you always have residual capsids that don't have a genome in there, and those would be useless for gene therapy, for example. So it's important that we know that ratio, and right now there aren't many strategies that are efficient to do that. So together with Affinité, we have embarked on a project to use their surface plasmid resonance technology to characterize AAVs. So SPR is based on the fact that substances attached to a chip will modify the light that comes in contact with it, and that quantitative information can be inferred from